Hello, everybody. Let's start our course with Introduction to Virology. In this first recording, in this first lecture, you will discuss we will discuss the nature of viruses, why viruses are studied, why do we need still study viruses, and we will also go over a brief history of virology. In second part, we will talk about detection and measurement of viruses and virus replication cycle. So let's start with the nature of viruses. However, before we go into the structure and nature of viruses, we have to identify difference between virus, virion, and provirus. Virus is very general term that refers to any form of the virus, whether it's virion, provirus, and so on. Virion refers to complete infective form of the virus, and provirus refers to integrated proviral DNA. So there are differences between virus, virions, and provirus. So let's see what is a virion. What does it make a virion, that infectious particle? Of course, every virus has viral genome, and we will talk later about that. It can be RNA or DNA. And this viral genome is protected by capsid, by the protein codes, which is called capsid. And this capsid can have either icosahedral symmetry or helical symmetry. Most viruses only are composed of the genetic material and the capsid, so-called nucleocapsid. However, some viruses have also viral envelope. Even though we call it viral envelope, it is derived from the host cell. And virus buds exits the cells, it pulls, it takes part of the cellular membrane and forms a viral envelope. However, inside of that viral envelope, there are viral envelope proteins, in most cases glycoproteins, which are specifically encoded by virus, and those are those key which recognize the locked receptors on target cell. So nucleic acid genome, capsid, and envelope proteins. This is a very basic structure you can find in any viruses. Now, what we have to know about the viruses is that they are obligatory intracellular parasites. What does it mean? If you see viral virion outside of cells, it does not behave as a live thing. It cannot proliferate, it cannot replicate, it does not react to other environment, it cannot metabolize anything. However, as soon as it enters target cells, it hijacks metabolic pathways of the target cells and proliferate and exerts all signs of life matter. So inside of inside of cells, virus can replicate and it can synthesize and forms a new variants. So why viruses outside of cells cannot replicate, cannot do much? Why they are obligatory intracellular parasites? Because they lack basic metabolic pathways that would synthesize biologic building blocks, such as nucleic acid, proteins, amino acid, and so on. They do not have also pathway which would synthesize the energy, generate ATP. So they cannot make protein synthesis. They also lack membranes, intracellular membranes, where all these mechanisms takes place. So viruses depend on the target cells to replicate and survive. When virus enters cells, it breaks down and releases the genome nucleic acid because it's uncoating. So major purpose of virus is to inject its genetic material in nucleic acid into the cell. it completely disintegrates, it collapses its envelope. And this is a major difference between, for example, bacteria and viruses. Even though some bacteria would replicate inside of cells, so let's see, like Mycobacterium tuberculosis infect and replicate inside of macrophages. However, it always preserves its shape. It preserves its membrane, it preserves its nucleic acid, it preserves all proteins. It just replicate, proliferate, it divides, but it does not disintegrate. It does not uncoat like viruses do. Now let's go back to genome. We said that genome can be RNA or DNA. It can be either single-stranded or double-stranded, either DNA or RNA. Yes, viruses can also carry double-stranded RNA or single-stranded DNA. They can also carry partially double-stranded DNA, such as hepatinoviruses. And this DNA can be either linear or circular. And we will talk a little bit more about that with each group of the viruses, particular structure and topology of the nucleic acid. So why we need to study viruses? Look, this is a graph which depicts the deaths by infectious diseases in United States from 1900 to 2000, and it shows number per 100,000 individuals. And as you can see, the death is decreasing except for Second World War, 1980 to 1920, when influenza pandemic, so-called Spanish flu, caused millions of lives. But afterwards, due to the precautious prevention, development of vaccines, deaths from the viral infection or infectious disease per se decreased. Now, exception was for early 80s, 1980s, when HIV/AIDS epidemic started. We don't see huge increase in death rate in USA on this slide. However, if you look worldwide, particularly in Africa, there was a huge spike in the death rate due to HIV/AIDS, not in US, but in Africa. So all this observation supported by this graph, which was taken, which was constructed for 2018 and 19, shows that major problem and major death rate is attributed to heart disease, coronary disorders. Second one is cancer. Still, second one's a huge death toll are attributed to cancer. But afterwards, the infection, influenza, and pneumonia, it's very negligible if when compared to heart disease or cancer. So this led to notion, why do we need to support the research for viruses in virology when they don't really cause such a harmful disease? Well, that was HIV/AIDS, but of course it affected mostly Africa. So this notion of not infectious disease being prevalent in US led still to cutting down funding for the virus disease research. All this view changed in 1920 during COVID-19 in pandemic. And this is from US. And this is a difference now. Yes, heart disease, number one, death rate, cancer, number two. But the third leading cause of death in US as in the world was due COVID-19. So it completely changed this picture of the death rate attributed to different conditions. And this look on 2021, the same, it persisted and number increased. So if we had enough resources and studied coronaviruses in more detail before pandemic, the vaccine and treatment would have been developed much faster. And this is a notion which I would like to get through, that even so, We don't experience, for example, pandemic of Ebola right now or other viruses, but it may happen. And knowing about those viruses, studying them in detail may help us, in the future to develop very efficient treatment and vaccine. So this table shows just some human diseases caused by viruses. Of course, you don't have to memorize them all, but the major one we will discuss in more detail. It's just for your information. Um, there are also fun facts about viruses. I know we usually attribute viruses to this pathogenic group, whereas they cause harm, disease, they harm us, they cause diseases, and so on. But if you look into world of viruses, actually, majority of viruses are non-pathogenic for humans, or even non-pathogenic. So we just focus on those pathogenic because they concern us, our health. So let's go to some fun fact. So we know that viruses can infect all form of life from vertebrates, humans, through insect, even bacteria. And they are most abundant form of life on Earth if we consider it as a life matter, which we know that inside cells they are. Why it is? I am sure you heard several times that the most abundant form of life are bacteria, particularly in oceans. And we have viruses, bacteriophages, which infect bacteria, and they are much more numerous than those bacteria. And if you align head to tail, all these phages, bacteriophages, they would cover distance of two times 10 to 21 kilometers, which is approximately a length of 2 times 10 to 8 of light years, 200 million light years. So all this, just if you align bacteriophages of the Earth, that's some fun fact, which is nice to know. Hurrah, bacteriophage. Okay, so in addition, why to study viruses? If you recall from general biology, numerous discoveries in molecular and cell biology are attributed to study of viruses. For example, DNA as a carrier of genetic information. Remember Hershey-Chase experiments? They were done with bacteriophages. Identification of promoters for eukaryotic RNA polymerases, enzymes which are involved in DNA replications. It's very important to see that viruses carry some protein and some enzyme, but they rely on cells for providing a lot of the enzymes, and some of them which are involved in RNA splicing, in promoters and RNA synthesis are provided by cells, and they can be distinguished from those which are actually synthesized and encoded by viruses, such as DNA replication. Also, viruses were first where we observed and isolated oncogenes, genes that are derived from proto-oncogene, and they play a role in the formation of the tumors. And there is this table, also very informative, which you don't, of course, need to memorize, but for your own interest, summarize some milestone in virology research. And surprisingly or not surprisingly, it started already in 18th century when virology was not even known, viruses were not known. And it started with Edward Jenner's first vaccine development. You recall he developed smallpox vaccine based on the observation of differences between people who were infected by smallpox and cowpox, those who milk cows. That follows by development of rabies vaccine by Louis Pasteur, also in 19th century, and from there many different discoveries. Rao sarcoma virus, first virus carrying oncogene, was actually observed at the beginning of 20th century by Peyton Rouse, and all this discovery leading to the discovery of HIV virus by Pasteur Institute scientists. Now, I'm returning back to question, are viruses alive or not? First, viruses were distinguished from bacteria because they pass through so-called filtration from these porcelain filters, which would catch bacteria. So it established, this methodology established that there are some infectious agents which are smaller than bacteria and they could pass through this filter. Next challenge to question whether they are alive was when the tobacco mosaic virus was discovered. It is RNA virus. And basically, what was very shocking and surprising that it has genes, it has proteins, but it was crystallized. It could be crystallized. Living organism cannot be crystallized. So this started this huge discussion: are viruses alive? And there is not clear-cut answer. In some textbook, you will see they are alive matter. In another, you will find they are not. My own definition is that viruses behave as the life entities inside of cells, but as non-life outside of the cells. And when we talk about history of virology, we cannot forget to mention so-called phage group. Around 1930s, a group of scientists decided to focus their study on the bacteriophages. Here is a nice picture of one of them. They observed, they knew that bacteriophages contain double-stranded DNA as a genetic material. They infect bacteria can be produced in huge amount, and they are mostly known infections for human. So they decided to study them as a wonderful biological object to lead to many discoveries. They predicted that their study might lead to discoveries, and they actually did. Not only this group of scientists mapped phage genome, they also elicited a replication cycle, developed plug assays. However, they also revealed genetic material of bacteriophages, whether up to that time, you remember, it was still question of chicken and egg. What is first, chicken or egg? Here, what is first, proteins or DNA? We know that DNA can replicate, but it needs protein to replicate. But protein needs DNA to be encoded, as this question was there in the center of biological discussions through the first half of 20th century. What is first? And several experiments concluded by Hershey and Chase, who labeled bacteriophage DNA by radioactive phosphorus and protein by radioactive sulfur, finally identified DNA as a genetic material of bacteriophages. Also, during the studies, Felix Darrell began work on so-called phage therapy, using bacteriophages to treat bacterial infections. And basically, this is still not accepted widely in US, but there are certain areas in the world when they use phages in case of antibiotic-resistant bacterial infections. Also, a study of viruses help to discover, as I mentioned before, some oncogenes. Rao sarcoma virus was first virus which was shown to carry oncogenes which are developed from proto-oncogene. Proto-oncogenes are genes in humans which actually play a very important role in cellular replication. However, some of them can be picked in by retroviruses, mutated and turned into oncogene. And when I talk pick up and integrate, also the study of viruses led to discovery of reverse transcriptase. We know that in humans, DNA can be replicated to DNA. DNA can RNA is synthesized of DNA, and protein are synthesized based on RNA. Viruses can actually convert RNA into DNA, retroviruses, with the help of the enzyme reverse transcriptase. Nowadays, viruses are also being used as a vector to express protein, as a vector for construction of the tumor cells, as well as vector for generating vaccines.